Mendel. Mendel and his contribution in genetics. G.J. Mendel, he was a famous mathematician. He was not a biologist. So he started his work on pea, garden pea. The name is Pisum sativum. So he worked on pea, and because of his excellent work, he showed what genetic is. He showed with certain observation and experiment, and that too mathematically. He was known as a father of genetics when he did that for 6 years, and he published the paper in the local newspapers and the local journals. He was not regarded as, or you can say his work was not attributed. But three famous scientists again from different corners of Europe, they found his work, and then he was regarded as the father of genetics. He chose 7 pairs of contrasting character. He chose P and 7 pair of contrasting character. See, this is a P. This is a gray and round P. This is a white and wrinkled P. There are 2 types of flower, violet and white. The different types of, say, this is a yellow pea, green pea. So he took 1 to 7 different pairs of contrasting character, and then he reached to certain laws, certain conclusions which are valid today, and he is the father of genetics. Now the main reason why he chose garden pea for experiment, that the pea has very short lifespan, around 2 to 4 weeks. The seeds produced are quite in a large number, so large number of seeds are produced. Then the self-pollination, the cross as well as self-pollination capability of pea, that was the reason he chose it. And also there are various contrasting characters that can be found in garden pea. So what was his law? There were 3 main laws: law of dominance, law of segregation, and law of independent assortment. What is the law of dominance? If the 2 alleles, what is this alleles? Alleles are basically the genetic content. We are talking about genetic content in organisms with a living plants, flora, fauna. So if 2 alleles at a locus offer, they differ, then one, that is the dominant allele. There are two allele, dominant and the recessive. So if the 2 alleles differ, then one with dominant allele, it determines the organism's appearance. Who determines? Dominant allele. The other, which is the recessive allele, has no noticeable effect on the organism appearance. This is the law of dominance. The law of segregation says if the 2 alleles, that is the genetic content for heritable characters, separate during gamete formation and end up in different gametes, so they segregate and they end up in different gametes. This is law of segregation. Then the law of independent assortment says that each pair of these alleles segregates independently of other pair of alleles during gamete formation. Monohybrid crossing. So one pair of, this is a pair, so one pair of contrasting characters, this was taken to cross 2 pea plants, and it was known as the monohybrid cross. So if you see this figure closely, this is showing that the monohybrid cross between a true breeding, this one is yellow pod, and this is true breeding green pod. So all the pods obtained was green in color. That is, when they are being combined, he found that all were green. This was capital G, the combination was capital G, small g. So the content was there from both these, but it was green. And the offspring obtained was known as F1 progeny. In the heterozygous condition, this is a pistil, this is a pollen. So when they are combined, or that they are supposed to be heterozygous condition, the parents are heterozygous here. So the phenotypically, what is phenotypically? Phenotypically is outside observable characteristic. while the genotypically is the inside genetic level characteristic. So if you see here, the parent are heterozygous, these two are heterozygous. So phenotypically, three purple, three purple flower were formed and one white flower was produced. But inside that, if you go genetically or genotypically, one homozygous, this one, two heterozygous, these two, and one homozygous recessive. This is dominant, homozygous dominant, this is homozygous recessive, and these two are heterozygous. All the three are homozygous. This is homozygous dominant, capital B, small b. Dihybrid cross. When two pairs of contrasting characters are taken to cross two plants, it is known as dihybrid cross. See, this is yellow round P and this is somewhat green wrinkled P. And these are the combinations. Say YR, this is small y, small r, and the combination at F1 was yellow round. It was this only. And when it self-fertilized, it can be any combination. So there are four possibilities here. These are four possibilities combination. So 4 into 4, it gives you 16 combination. These are all the 16 combination. So phenotypically means if you see outside observable characteristic, the ratio was 9:3:3:1. What is 9? 9 are round yellow. These are round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellow. Then there are 3 round green. 1, 2, 3. 3 round green. Then 3 are wrinkled yellow. 1, 2, 3. 3 wrinkled yellow. And 1 was wrinkled green. But the genotypic ratio was somewhat like this: 1:2:1:2:4:2, 1:2:1, like this.